So thank you for inviting me, Joel. I've just put together this talk a week ago, so it was on the fly. It's a little dangerous. This is a dangerous talk, and the reason is that three of the four people that I'm going to be talking about their work are in the room. So Johan Gunjansson, Michelle Gillier, and Jonathan Barker, is he still in the room? I'm going to be talking about some of their papers. Basically, my task went to review posters at the AAD to hot topics. And for me, I'm an old scientist. For me, the most hot topic in psoriasis is a methodology called single-cell RNA sequencing. So I'm not personally doing this work, but I'm an old scientist and I understand it, and I'm really excited by it because it's offering insights to pathogenesis of psoriasis in ways we've never been able to do. Now, most of you have seen the old picture, right, of IL-23 being made by APCs leading to T-cell, making IL-17 leading to keratinocyte activation. And then we have the feedback loop of the keratinocyte cytokine production. So that's been around forever. So really, this technique in just the last year has challenged that old slide, if you will, and made it much more complex. So the message here is that psoriasis pathogenesis is much more complex than we expected. So the first, I'm going to take you through 4 papers, and it's a high-level view. There is so much detail in these 4 papers that it's impossible for me to talk about them in detail. So I'm going to give you the high-level view of these exciting papers that have used this new technique, single-cell RNA sequencing. So the first one is from Johan, who you heard speak of earlier. And it asks the basic questions that we always ask: which cell types and which cytokines are the main drivers of psoriatic inflammation? And I told you what the paradigm was previously. Dendritic cells making IL-23, stimulating T cells to make IL-17, stimulating keratinocytes to make a whole bunch of things. And then how do the numerous cell types and cytokines and psoriatic lesions interact with one another? So that's another thing with this technique is how the interactions are occurring, which is novel, which we have not really experienced before now. So it's opening new windows. So here are some of the figures from this paper from last summer. And in the upper left, we can look at now tissues, and we can tell where the cell types are. So each color in the upper left is a different cell type. We've got keratinocytes, smooth muscles, fibroblasts, and so forth. In the middle upper panel are different types of T cells. So this is way beyond Th1, Th2, Th17. We've got naive T cells, stress T cells. Number 1 and 2, we got Tregs, cytotoxic T cells, TC17s, gamma T cells, and so forth. And we know now, we've defined these cells, or Johan has in this paper, based on their gene expression. So not only we know that they're T cells, but we know what genes they're making. That's the essence of single-cell RNA sequence analysis. We can identify gene expression within individual cell types and in the location within the psoriasis lesion where those cells are and what genes they're expressing. And so one of the major findings in the lower panel, you can't really see it in detail, is that Johan found that CD8 positive TC17 cells, this one type of T cell, is the major source of IL-17A in psoriatic lesions. That's an important finding. We know IL-17 is important, right? And this in particular he has said that CD8 positive TC17s are the main cell type making IL-17. Another major finding is the role of IL-36. So IL-36 we know is important in pustular psoriasis, and we have a drug now, spesolimab, that blocks IL-36. But what about IL-36's role in plaque psoriasis? And so Johan has found that IL-36 in these studies amplifies IL-17A responses in the supraspinous epidermis of psoriatic skin. So here's a role for IL-36 in plaque psoriasis, not just pustular psoriasis, amplifying the IL-17 response. The other major finding, kind of a surprise finding, is that there are a type of fibroblast in psoriatic lesions, SFRP2. I'm not sure what that stands for. I had to look it up. Inflammatory fibroblasts that transition from a pro-fibrotic state to a pro-inflammatory state within psoriasis lesions. So now we have fibroblasts, which was never in our pictures. Mm-hmm. Never in our pictures did we talk about pro-inflammatory fibroblasts playing a role. And here's Johan talking about those cells. So really fascinating results. I'm going to talk about a second paper now, and this is from Michel Gillier's lab. Is he still here, Michel? Okay, he was here earlier. This one is about how do TH17 cells, TC17 cells, come into the skin, and how do they start making IL-17? And this is a fascinating paper because, for me, it talks about the interaction between T cells and keratinocytes in psoriasis. And so what he did in the upper left, in the very upper left, is the TGF-beta expression coming from keratinocytes. And the next panel to the right is IL-26 expression coming from T cells. And then in the left middle is showing an overlap of TGF-beta and IL-26. And what he's shown is that there is co-localization of keratinocytes making TGF-beta and T cells making IL-26, and the co-localization is occurring in the epidermis, and it's not occurring in AD lesions, atopic dermatitis, but just psoriasis. So what he's saying here with this paper is that there are IL-26 positive TH17 precursors that come from the blood, they go into the skin. The IL-26 from these T cells induce the keratinocytes to make TGF-beta. The TGF-beta then crosstalks back to the T cell and turns the IL-26 positive precursor into a full-on IL-17 producing TC17. This T cell coming in, it's not making IL-17, but it gets a signal from the keratinocytes, and then all of a sudden the T cell starts making IL-17. So this crosstalk, fascinating stuff. We've never known anything like this before. Number 3, here's another paper. This is a paper about what happens after you block with IL-23. What happens to single cell seq analysis in the first 2 weeks after starting risankizumab, an IL-23 blocker? What are the changes that happen first? This paper just came out in January. As Jonathan Barker was involved with this. So what you have in the top left is patients on psoriasis who start to get better right away after a single dose of risankizumab. They start to get better day 3, day 14. And they were able to look at the changes in the keratinocytes over the first 2 weeks. So the genes start to change in the first 2 weeks in keratinocytes, in T cells, in myeloid cells. But the big finding is on the right. The biggest change is in these inflammatory fibroblasts. Again, fibroblasts. Inflammatory fibroblasts are one of the things that changes the most, putting them from an inflammatory state to a non-inflammatory state in the first 2 weeks of risankizumab. So kind of things we just have never seen before and never have known about. And here's a schematic, adding now in fibroblast, saying that basically you get this IL-17 making them into pro-inflammatory, and basically with treatment you can calm down the inflammatory fibroblasts and turn it back into a normal fibroblast, and it happens very quick within the first 2 weeks of treatment. All right, and the last one is another example of IL-23 inhibition. And in this case, the investigators looked at tildrakizumab-treated patients, and this paper just came out in February in Science Immunology. And they looked at people who did well on Tildra versus people who did not do well on tildrakizumab. So we know that Tildra is not quite as potent as a IL-23 blocker compared to risankizumab, so there are some non-responders with Tildra. So they compared the immunology, if you will, on a single cell level of people doing well versus not doing well. And I don't know if I have a pointer here, but there's a population shown in the middle panel here of T17 cells, and they go way down. I'll just tell you the answer. They go way down in responders to tildrakizumab. And the non-responders to tildrakizumab, the T17 cells are persisting. They're staying there. And they still are causing the whole IL-17 induced gene expression pattern in the folks who are not doing well on that particular drug. So it's an example on a cellular level of what's happening in non-responders versus responders. You have this persistence of the T17 cells in people who are not doing well on the drug. And then one last little plug, we, along with Johan, we have used single-cell RNA-seq to look at patients in our knockout study, and we'll be presenting this data on Saturday morning at the late breaker session. And in this case, we use this type of analysis to look at resident memory T cells. So we focused on resident memory T cells in this study, and hopefully you'll be able to come to the late breaker session on Saturday. So with that, thank you for your attention.